All right, hello everyone. I am going to take the time to just go over the highlights of different brain imaging techniques because I know sometimes they can all sort of run together for you. So this won't cover all of the material from the chapter or the SGA. It'll really just hit at the highlights. So we're going to talk about imaging the brain's activity and talk in particular about the different techniques. The one thing we won't talk about that your chapter goes over is brain stimulation. It is really helpful, but not something we're going to focus on in this particular mini lecture. So there are three techniques for recording the brain's electrical activity: single cell recording, EEG, and ERP. And going with ERP is a newer technique called MEG as well, or MEG. So we'll touch on that briefly. So single cell recording is not usually done with humans. It's not that practical. So it's more often done with animals. It's a way that you can look at individual neuronal activity. You can do multiple neurons simultaneously. So these are action potentials, and then you can see how quickly these actually occur. And the rate of firing can tell us some information about. Again, not practical for humans because you do have to implant an electrode in the brain next to the neuron, but we can do it with animals in the laboratory, which, again, the ethics of that are definitely up for debate. Certainly something to look into if you're interested in that yourself. EEG, or electroencephalographic recording, was developed in the 30s. So this is one of our first imaging techniques. So it records the brain waves, essentially. So we often use it for sleep studies, anesthesia, brain function, and it's diagnostic, particularly for disorders like epilepsy. So this is what an EEG looks like. They're attached to the skull. The electrodes, they're not implanted like you would for single neuron recording. And then this is what an output looks like. So you've got a pen, although a lot of times now this would be replicated digitally, and it shows the electron activity occurring within the brain, which are essentially the neurons firing. So these are the different patterns of brain waves that are associated with particular behavioral states. So you have the beta rhythm is your awake state. Alpha waves are calm and rescue. Theta waves are drowsiness, and then delta waves are deep sleep. So these are what those particular waves look like. So again, beta is when we're awake. Alpha is when we are relaxed, our eyes are closed. Drowsy happens when we start slowing things down with C waves. Then you have asleep, your delta waves, and then deep sleep. You have these slower, high amplitude waves, and then finally, if you're in a coma, there's even further slowing. Now, the really interesting thing is if you're in rapid eye movement sleep, REM sleep, where we do most of our dreaming, you'll actually look most like beta rhythms, most like you're awake, as opposed to the others. So as I mentioned, you can use this to diagnose epilepsy. We'll go into epilepsy a lot more later in the semester, but basically you can have petit mal or grand mal seizures. That's an oversimplification. There's lots of different types, but we'll start with that now. Petit mal is essentially you space out, and grand mal is what we think of when we typically think of seizures. So the convulsing, the falling down, sometimes loss of consciousness, certainly loss of bodily functions, for example. So EEG helps us figure out where those problems are happening. And so this is an example of what that might look like. So we could do this during and before and after a seizure and see where in the brain it seems to be located. ERPs, you look at a brief change in a slow wave EEG signal in response to some sort of stimulus. That's an ERP. So you present the stimulus repeatedly and you average the recorded responses because it'll be slightly different every time, and you want to make sure it wasn't a fluke that they were responding to something else. So here's an example of what the first response might look like, and then how you would average it over numerous responses. So for example, maybe you give an auditory stimulus and then look at how people respond to that. You would also do this to a visual stimulus as well. Magnetoencephalography, or MEG, or sometimes called MEG, is another example of a technique that can be used that's similar to EEG. So it records the magnetic fields of neurons rather than the electrical activity. So it is high resolution, but because of that, it's pricier, so not necessarily used. And one thing I should say is in the videos for this section, there are video examples of what all these different techniques look like, which I think is even more helpful than me talking through this. So be sure to check those out. The first way we actually image the brain, and not just its activity, was just with an X-ray, just like we would any other part of the body. And then that gives us a static 2D image. The way that we have adapted that is to try to get more of an overall picture of the brain using different techniques. So we're going to talk about those in a lot of different detail here. So computerized tomography, or the CT scan, which also is often referred to as a CAT scan, passes X-ray beams through the brains at different angles to get images essentially at different slices of the brain. It's like when we sometimes do an autopsy of someone who had brain pathology, we will sometimes actually physically slice the brain. We obviously don't want to do that to someone who's living, so instead we can do it with X-ray beams. So it isn't as accurate or high resolution as some of the other techniques that we'll talk about, but you can see major fissures, you can see the ventricles, which we'll talk about on the next slide. So the ventricles are these dark patches in the brain. Everyone has those, the things that kind of look like kidney beans on this one. It's normal to have those, but you can see whether it is enlarged, right? You can also see if there are dark spots where the ventricles aren't supposed to be. The ventricles are fluid-filled. They help with things like balance and homeostasis in the brain. So here you can see a lesion on this CT scan, and then you can see where that lesion would actually be. A lesion is an injury, essentially. We can also do what's called dynamic brain imaging now. So this is where we essentially, instead of taking a picture, we take a video of the brain as we analyze it. So we can do this with PET or PET scans, with MRIs, and there are other ways we can do this as well. So positron emission tomography, or PET scans, uses a subtraction method to look at brain activity, but does so in a dynamic way. The computer's going to reconstruct variations in the density of the flow of particles. So here you see what the PET scan looks like. So you've got this radioactive agent that's injected, and then that is going to interact with the brain molecules, and that allows the PET scan to look at the activity in the brain. So here's an example of how we can get that activity at different levels of the brain. And here you can see where we would have someone look at like stimulus and then not, and you can see the difference here. And we would look at that for individuals if we were doing this diagnostically, but in research over time, we actually will average that of individuals to get an overall view of which parts of the brain might light up while you're looking at a particular stimulus or doing a particular activity. Magnetic resonance imaging, or MRI, is something that's used all over the body, but in particular for the brain. It uses a large magnet and radio frequency pulse to produce an image. And this image can either be static, or you can, again, like PET scan, do this while someone does a task. Now, MRI, one big caution with it is if a person comes to you, say to the ER, unconscious, and there's no one with them who knows their health history, it can potentially be dangerous to do an MRI because it's a big magnet. So if that person has a screw in their knee, has a pacemaker, any number of things that might include metal in your body, it's not going to like tear it out, but it certainly will move it around through your tissue, which obviously could be potentially really problematic because you've got this big magnet with the little metal in the body. So in that case, you would do a CT scan first until you knew the full medical history. Here is what an MRI looks like, and here's what the imaging from it would look like. Now, one thing I do have to say is with MRI, some people do get quite claustrophobic because it is a tube that you're going into. So a lot of places have started implementing techniques to try to help with that. There are open MRIs that you can use. Some places will have you wear headphones, play music, project imagery to try to distract you from this. Now, fMRI, or functional MRI, is where you would use the MRI as a video, essentially, where you're recording the activity in the brain as someone engages in a task. This is done for a lot of different psychological research now. So I've seen MRI studies, and I believe there's a video posted where they have people read passages or recite monologues. It can also, again, be diagnostic, and here you can see where they're just turning on a light on and off and seeing how the brain reacts to that. So in comparison of these different techniques, here, you know, again, single cell recording, too invasive to use with living humans at this point. EEG gives us this non-invasive, inexpensive, but only kind of gives overall patterns. It doesn't image anything that might be going wrong in the brain, like a bleed, like a tumor, like a lesion. X-ray gives us that quick static shot, just like if you broke your arm, for example. MRI and fMRI are high resolution. CT is that nice go-to if we don't have medical history. And then again, that PET scan gives us the biochemical status of the brain. PET and fMRI allow us to see the brain as it actually reacts to something. I hope that this helped clarify things a little. Again, check out the videos on Blackboard. I think that they'll be even more helpful.